Hello, this is Christine Deal, and we're going to be covering Chapter 7 today, Sex Chromosome and Mitochondrial Inheritance and Disorders. Okay, we're going to start with the sex chromosome disorders. These are with the X and the Y chromosome, so these are different than the autosomal disorders. Okay, so again, the X and the Y chromosome is what we're going to start with. So there is a condition where there are extra X chromosomes with trisomy X, with one extra X. It's really not that phenotypically obvious. The main expression is that these individuals tend to be taller and taller than their siblings. It is a common occurrence. It occurs in both males and females. And as I mentioned, that single X is associated with few phenotypic manifestations, but the more X's that one has, the more problematic it is. So trisomy X karyotype shows 47 because you got that extra chromosome, XXX. All races are equally affected. It happens in 1 in 300 to 1 in 400 births, so that's fairly common. Normal female phenotype, and again, as I've mentioned, the height is greater than average and greater than that of their siblings. There might be a slight delay in language and motor skills, or it may be barely noticeable. Fertility is unaffected. But again, when we get into more extra X's, that's where we get more severe phenotypic expressions. Again, all races are equally affected. The incidence is not known with these. Tetrasomy, they are taller and they have cognitive disability, so they have greatly reduced intellectual function. And then when you get to pentasomy, or 5X's, that is a little different because that's short stature and not tall stature, and a severely reduced intellectual function, cleft palate potential, coarse facial features, hypotonia, microcephaly, hypertelorism, and heart defects. So this certainly is a more severe expression and condition. So here's a picture of a tetrasomy with 2 extra X chromosomes. So here you can see hypocanthal folds, flat nasal bridge, small mouth, cleft palate, potential delayed teeth, or absent teeth, enamel defects, and again, the tall stature. But to look at this picture, you might not, it's very subtle, but you do tend to see these expressions. Now, when you get to pentasomy X, you can see here in this karyotype, there are those 5 X's. This picture is of an affected 11-month-old. You can see the cleft palate there. They also tend to have, again, also a flat, broad nose, associated with short stature, poor muscle tone or hypotonia, the small hands and feet. These individuals are more severely mentally deficient with developmental delays. This can be misdiagnosed as Down syndromes, so it definitely needs some analysis there because they have some similar features. Okay, and sometimes there is an extra Y, okay, and that is known as Klinefelter syndrome. So you have 47XXY. So these individuals are genotypically male, okay, but they may have feminine features that may not be noticeable until may not be diagnosed until they hit puberty or they have delayed puberty. All races are equally affected. Incidence is 1 in 600 male births because they are genotypically male. Normal infant and childhood male phenotype. However, I did service coordinate when I was in the public health department for, she was a toddler at the time, but she was assigned male at birth, and then because she had ambiguous genitalia, so she was assigned male, but then the family made the choice to change her to a female, as far as how they referred to the child. and that was in infant and toddlerhood. This is caused by maternal and paternal non-disjunction associated with advanced paternal aging, but has occurred in offspring of young adults. So this particular toddler that I service coordinated for was the offspring of two teenage parents. So even though I know the book says it's mostly associated with advanced parental aging, it has occurred and can occur in young parents as well. So the features of Klinefelters is that they tend to be taller than average. The legs are long relative to torso length. They have low testosterone levels because of their extra X, high FSH levels, loss of secondary sexual characteristics. So many times, like Klinefelters isn't identified until they start to hit puberty. Okay, so small testes and penis, gynecomastia, decreased libido. Okay, they are infertile. Osteoporosis because of the just the fluctuation in the hormones, the different hormone levels. They tend to have lupus. Of course, all this is diagnosed later in age. They may not be, like it says here, right here, it may not be diagnosed until puberty and testosterone can be used for treatment to help with fatigue, to build the muscle, and to prevent the gynecomastia that occurs from having that extra X. Or I mean, I say extra X, I mean, it's XXY, so the Y is what is making them male, but they're male with two X's, which it feminizes them, which isn't noticeable until they get to puberty. And they do not have a uterus. Okay, so in adulthood, there are some Klinefelter associated features, and that includes disorders like thyroid disease, diabetes, lymphomas, germ cell tumors. IQ is slightly lower than that of siblings. They do have delayed language and reading skills, delayed walking and motor skills, which was the reason why I was a service coordinator for the toddler, because they assigned her a female, but genotypically a male. She had gross motor delay, okay, delayed speaking, walking, motor skills, and so I was service coordinating for her physical therapy and speech therapy. Klinefelter's has a higher incidence of autism spectrum disorders and a higher incidence of ADHD. Okay, so there is extra Y chromosome condition. So two Ys instead of an extra X. Okay, so this is a case of where you have one X and two Ys. So again, they are male. They are. It occurs from a paternal error in meiosis 2. Affects all races equally. The incidence is 1 in 1,000 male births, so a little bit less common than the Klinefelters. The only phenotypic feature is tall stature. Fertility is unaffected. Increased incidence of 47 XYY among offspring. Okay, so this can be passed down. It is associated with severe teenage acne because they have the extra Y, they have the extra testosterone. Also, slower motor and language development. Okay, so now we are going to go into monosomy. And a lot of this is straightforward, and your book is really good about discussing all of these disorders. Make sure that you just have a way to organize all the different disorders, so you can keep them clear on what they are and what the manifestations are. But monosomy involves missing a copy of one chromosome. Okay, so it can result from non-disjunction, okay, which allows one gamete to retain both chromosomes of a pair and one gamete to have no chromosomes for that pair. Non-disjunction is more common with advancing maternal age. However, again, I don't know. I have an old college friend who had her third baby when she was in her mid 20s, and her third baby was affected with Turner syndrome, and again, she was not of advanced age. So even though it's more common, I just want to make it real clear that it can occur. Okay. It can also result from gamete formation in a person who is a balanced translocation carrier. So I don't know if they were tested for that, but this certainly was a young couple, my friend and her husband. Okay, so monosomy X, Turner syndrome, 45X. Okay, so they have one less sex chromosome, and Turner syndrome is the most common chromosome problem conceived. According to your book, most conceptions are lost as early miscarriages, and the live-born infants are thought to have undiagnosed mosaicism. Okay, and we talked about mosaicism in chapter 6. So there is mosaic Turners in addition to full Turners. But this individual, my friend's daughter, was not mosaic Turner. She was a Turner syndrome. And they are often diagnosed by prenatal ultrasound or at birth. And they are characterized by having a webbed neck. Here's the incidence. You don't necessarily have to memorize these incidences. This just gives you an idea as you are studying this to realize, to see kind of like, well, how rare is this? How common is this? It's associated with many different features. Not every person has all of them. It used to be thought that Turner syndrome was decreased cognitive functioning, decreased IQ, but more recent studies have shown that intellectual functioning can be the same as general population. So there's just a varying degree of effects, and especially if it's mosaic Turners as well. Just like mosaic Down syndrome can have Down syndrome features, but have no cognitive effects because the cells in the neurological cells in the brain aren't affected. So there's a varying range there. Individuals share variations in skin, hair, and eye color with family members. Okay. So there's no, like, they tend to have blonde hair versus, so it's not like with Angelman's. Okay. So they still tend to have some family features. Some consistent features is they do have short stature and decreased growth rate. There's no adolescent growth spurt. The average natural height of someone with Turner syndrome is 47 inches. Pedal edema is seen in newborns, and that's part of that lymphedema. Frequent but variable features are the neck webbing, which is very classic for Turners. It makes it very specific for Turners. Kyphosis, scoliosis, and osteoporosis. Now, with the decreased growth rate, they are given, it is federally approved. The FDA has approved growth hormone for these individuals to receive, but the growth hormone is not effective, really. If it's not a true growth hormone deficiency, then the rate of growth is not going to be that effective. They might get a little taller, but they're still considered, they still have very short stature even with the use of growth hormone. So here's a picture of some characteristics. Again, so you can see the webbing of the neck that occurs in both these pictures. And also, again, keep in mind mosaic Turner syndrome, varying degrees of expression. They're short stature with infertility, but not always the webbed neck, and maybe only a slight mental deficiency, or maybe just a learning, slower to learn, or some learning deficiency. But these individuals have completed college degrees. So here's a picture of the infant with Turner syndrome with that pedal edema that we discussed. Here are some more Turner-associated features. So, this is pretty straightforward. Most specific, notably, I want to talk about the aortic malformations and coronary artery disease because these individuals can be treated, and many parents do choose to treat them with growth hormone. And growth hormone, too much growth hormone, can magnify these cardiac effects. Okay, because we know too much growth hormone can cause cardiomyopathies. Okay. So they need to be closely monitored if they're on growth hormone. All right, also more Turner-associated features: infertility, underdeveloped uterus, hyperlipidemia, which is also going to be a concern if they have growth hormone added to their treatment. Life expectancy is lower than average, mainly because of those cardiac effects. They have absent or delayed menses because they have underdeveloped ovaries. Okay, and with that, poor breast development. Okay, so now we're going to move to Fragile X syndrome. So Fragile X syndrome is the most common genetic problem leading to reduced cognition among males. So that involves silencing expression of the FXMR1 gene, and that gene is responsible for brain development and maintenance of neuronal synapses. Okay, so in classic Fragile X, the FMR1 gene coding regions are normal, but the gene's expression is silenced by large numbers of the trinucleotide sequence of CGG being repeated within the non-coding regions of the gene. Okay, and so that leads to silencing. So when you have the gene that's silenced and it's responsible for brain development, then it goes to reason that you would have delayed cognition, mental delays, mental deficiencies, okay, because you don't have you don't have proper cognition and neuronal synapse formation. So again, just to recap, silencing results from the excessive trinucleotide repeat sequences and methylation within the non-coding regions of that gene on the X chromosome. Okay, and when it's silenced, it cannot function. So be sure you know the function of the FXMR1 gene. So the degree of expression in Fragile X syndrome is related to the number of repeat sequences. So less than 50 usually has no expressed problems, but the more you get, obviously the more severe the syndrome is. So women with 50 to 200 repeat sequences are premutation carriers. And so then they can transmit this change to their children in an X-linked recessive manner. So the problems occur when the number of repeat sequences increase by expansion. So be sure you understand the concept of expansion and anticipation. And just as a side note, the term Fragile X comes from the tendency of the X chromosome to exhibit a small break or a gap at the end of the long arm, okay, of the chromosome. It occurs in 1 to 4,000 male births. It can occur in females, in about 1 in 8,000 female births. It is, again, it's the most common chromosome problem leading to decreased cognitive function and intellectual capacity in males. The difference of expression in females is likely the result of random X inactivation because females have two X's that kind of gives them some more protective mechanism, so to speak, and so they could have the random silencing of that Fragile X and not be affected or have result of random X inactivation. So they have that other X to kind of protect them from more severe effects. So expansion and anticipation. So expansion of repeat sequences tends to increase with each succeeding generation, which is the anticipation. So it's often initially diagnosed in a male child in a family with no history of the problem because you have, all these repeat sequences and it's got to get to be around, over 200 for it to then show phenotypic expression. So that is why these individuals can be born to no family history at all, because it's been hidden. So it's usually transmitted by an unaffected grandfather, which is referred to as the normal transmitting male, to all of the daughters. And then the daughters are premutation carriers, which we discussed on the previous slide, with then disease expression in a grandson. Because with each generation, those numbers, those repeat sequences increase. So all mothers of affected sons are carriers who have a 50% risk of transmitting the mutation with each pregnancy. Okay. So the higher the numbers of the repeat sequences, the greater the methylation, which then equates to increased severity. So associated features, autism spectrum disorders, poor balance and coordination, reduced cognition, as we've mentioned, because we know what the role of that FXMR1 gene is, poor verbal skills, anxiety, poor tolerance of change, long narrow face, large ears, a prominent jaw, and then a high arch palate. They have got a picture of a Fragile X child coming up here soon. More associated features, of course, they have some gross motor delays, some other tremor occurs with aging. Again, there's sort of a varying expression here. So here we go. We have a child, a school-age boy with Fragile X syndrome. So you can see the long, narrow face. Okay. There are also some Fragile X syndrome associated syndromes. So there is an associated primary ovarian insufficiency syndrome, which occurs in some premutation carriers, and this is where you have a female with an early onset of menopause before age 40. Okay, so that's suggestive of having this syndrome of being a premutation carrier. Menopause before age 40. Yes, it is sort of one of those symptoms of a premutation carrier. Also, there's associated tremor ataxia syndrome, where they get progressive ataxia and tremors after age 50, and that usually occurs among people with 50 to 200 repeat sequences who do not repeat Fragile X syndrome. So over 200 repeat sequences is where you have the phenotypic expression, but there are people who have a lower range, less repeat sequences, so they don't phenotypically express it, but they are still at risk for tremor and ataxia later in life. Okay, and so we're going to move on to genophenotype gender mismatch. I kind of went in a different order than your book does because I wanted to talk about all the disorders together. I don't know. I'm not necessarily going to, this is just something that I want you all to be aware of that occurs genetically. It's not a main focus, but I think it's for the exam, but I think it's an important thing, an important condition to be aware of. So some people have a natural genotype of one gender and the phenotype of the other gender. So it's known as sex reversal. And that is a purely genetic condition. Okay, so this issue may not come to light until the person enters puberty and development does not proceed as expected. Okay, so most often the difference between gender, genotype, and phenotype is related to embryonic tissue that is insensitive to the normal hormonal influences or the hormones responsible for directing the specific tissue development are not present. So both issues are genetic in origin. So they could have the genotype of one sex, phenotype of the other. So after commitment in early embryonic life, the cells destined to become gonadal tissue can develop into either a testis or an ovary, depending on the chemical, the hormonal environment in the female. So it's referred to as a bipotential gonad. So regardless of genotype, the precursor tissues for male and female genitalia are both present. So this is discussed in your book between pages 135 to 137. So there can be a female that is, well, there are XY females, okay, because remember they're bipotential. So they can have an androgen insensitivity syndrome, okay, and so the androgen receptor gene is located on the X chromosome, not the Y. So there could be just some insensitivities to certain hormones that is a component of this genotype phenotype gender mismatch. So just be aware that is a potential and that can occur. So when the genotype allows production of the appropriate hormones and other protein products, the precursor tissues for the correct gender are enhanced and the ones of the other gender are suppressed. So if the right hormones or influencing proteins are not present because of a gene mutation, like if you've got androgen insensitivity, then the wrong tissue may develop, for example. So even if the right hormones and influencing proteins are present, again, like I said, if the receiving tissue lacks the receptors for the substances, they will not respond to those inducing substances, and they will not develop the same phenotype as the genotype because they will not develop into the correct genitals for the genotype. Okay. And the opposite tissue may develop to some degree, but not completely or correctly. Okay. So that is what is referred to as the gender mismatch. They actually have both like gender reversal. Okay. So that is a genetic condition. Okay, so now we are going to move into mitochondrial inheritance and disorders. Okay, so remember the mitochondria are the powerhouse of the cell. So they are organelles in the cytoplasm that generate the high energy needs of the cell. Okay. So they are most abundant in tissues with high metabolic rates, okay, like cardiac, respiratory. They contain a small amount of primitive DNA that has few genes, all coding for proteins needed to produce ATP, which is the energy. Mitochondrial DNA is known as mtDNA, and this is derived completely from the female lineage. The male does not supply any mitochondrial DNA because the ovum is the largest cell and it has all the mitochondria in it. The sperm does not contribute any mitochondria to the zygote. The only mitochondria that's present in the sperm is in the tail, and that helps it for motility to fertilize the egg. Once the egg is fertilized, the tail falls off. There is no mitochondria exchanged from the male. Okay, so mitochondrial DNA replicates separately from nuclear DNA and has few repair mechanisms. Mitochondrial DNA mutations reduce cell function by limiting the amount of ATP available. So if you need energy for function and then you have a mutation where you don't have that, then that's going to cause a deleterious effect. And again, I've mentioned and explained that all mitochondria are inherited from the mother at conception, and that the mature ovum at fertilization is large, has a huge amount of mitochondria, and that the sperm does not. And this is a picture from your book, the first 5 rounds of cell division for a zygote. Nuclear DNA is replicated and the numbers of cells are increased, but the size of the ball of cells does not increase. There's no crossing over. Okay, so really that's all this picture is there to depict. And again, here's, not again, but this is a picture of the, obviously the sperm, and it shows that the tail piece containing the mitochondria that then falls off. And so there's no mitochondrial exchange from the male. So this I've already explained. There are a few genes in nuclear DNA that code for proteins involved in mitochondrial function, ATP production, but they are small and seldom mutate. So the vast majority of mitochondrial problems arise from mutation in the ova mitochondria. So the disorders of mitochondria occur at a rate of about 1 out of 10,000 live births. Problems most frequently noted in tissues requiring high performance of mitochondria, or those tissues that have high ATP or energy needs, which would be the musculoskeletal system, the cardiovascular system, and the neurological system primarily. Expression of problems are variable because of uneven distribution of affected mitochondrial DNA. Okay, so mitochondrial disorders are discussed in your book. Leber's hereditary optic neuropathy is one. It is very tissue specific. There is bilateral progressive vision loss that is expressed in early adulthood. And so here I've got a picture of healthy vision and then what vision of someone affected with this has. Just to give some idea of what they see. There's also myoclonic epilepsy with ragged red fibers. Affects many tissues. That expression begins early in childhood, and that progresses to include ataxia, muscle weakness, dementia, hearing loss, blindness, and cardiomyopathies. And really, the main thing we want you to know about mitochondrial disorders, number one, we want you to be aware that they exist and that they derive from the mother, and that they affect tissues that require high amounts of ATP. There's also mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, or MELAS. This affects many tissues and organs. It begins in middle childhood, recurrent headaches, seizures, muscle weakness, could have intermittent one-side paralysis, and then by adulthood, this is definitely a more severe one. In adulthood, manifestations progress to impaired motor ability, reduced vision, hearing, cognition, and diabetes. Okay, and so that wraps up chapter 7. If you have any questions, please feel free to text, email, or call me. And I hope that you have a good rest of your day. Thank you.